This video is going to start you right after the dihybrid cross in the genetics notes. It starts on page 3 unless you've changed some of your pagination. It starts with define a pedigree. Pedigrees are essentially family trees that look at a disease or a trait in a human population to find out whether or not that disease might be dominant, recessive, genetic at all, or is it something that might be environmental. We do pedigrees because you can't just breed people together to see how traits are going to be passed down. So pedigrees can be useful for finding out, for example, that freckles are the dominant condition or the straight hairline is the recessive condition, like these pictures are trying to show you. Next up, what we expect you to be able to do in both lecture and lab is recognize, based on a pedigree, whether or not a condition is dominant, recessive, or sex-linked. We've already talked about what dominant and recessive means, so I'm going to skip down to sex-linked for just a second. If a condition is sex-linked, that means it's carried on the X chromosome. Since guys only get one copy of the X chromosome, that means they only get one chance to get a good copy of that gene. So the condition is more likely to pop up in males than it is in females. Sex-linked conditions are a type of recessive, but it's a special subtype that gets its own classification. We're going to look at a few different pedigrees to make sure you can understand and recognize those inheritance patterns. Some of the first really basic things that you need to make sure you understand is the generations are labeled with the Roman numerals across the left of the pedigree, and so there's 3 generations present here. You could kind of consider this like parents, kids, and then grandkids if that helps you to picture what's going on. Next, each person is labeled with an Arabic numeral, so this is individual 1-1. The numbering always starts over on the next row, so that's individual 2-1. So this helps you talk about people, because when you're looking at pedigrees from a medical standpoint, you don't want to know the names of people. You have to try to keep things blind, and so numbering them like that just helps us to talk about them. Next up, you'll notice there's boxes and circles. The boxes are males, the circles are females. And that's going to hold true for all the pedigrees that we look at. So boxes are always boys. If it helps you to remember it, box and boy starts with B, and so they go together. Anytime you see a person who is shaded in, like individual 1-2 or 2-3, that means they're affected by whatever the condition is that you're looking at. If you have a line that connects 2 people straight across, like that line there, that means, and I'm going to be euphemistic here, that means that they're married. What it means in real life is they had sex, they made some babies, but it means they're married for our purposes. They'll have a line that comes off of them that takes you to all the babies. So all of these guys that are connected with these lines, those are the children of the first generation. So they had 3 kids. Individual 2-2, 2-3, and 2-5 are their biological children. Individual 2-4 married in, so this is a daughter-in-law is how that's going to work out. If you have a line that goes up and over to connect 2 people, then that means they're siblings. So line straight across, married, line up and over, siblings. Next, when you start to first look at a pedigree, what you want to do is figure out who has the trait and who doesn't. So remember, everybody who's shaded in, they have it. If the trait pops up in every generation, and if a kid has it, at least one of the parents has it, that means that condition is dominant. And so this would be a dominant genetic condition. This pedigree, on the other hand, as you look at it, notice that the only 3 people that are affected within this family are all boys. That is a really strong piece of evidence that that is a sex-linked condition, otherwise known as a sex-linked recessive condition. Next, this is the pedigree that's actually in your notes. First thing we want to know is, what's the inheritance pattern? So the disease is popping up in every generation. However, there's a really important note to make here, and I'm going to go ahead and label our generations to help us talk about them. Individual 3-1, I want you to notice that she has the condition, which we're going to say that this is about cystic fibrosis, but neither of her parents has the condition. That piece of evidence that you have a kid that has the condition but neither parent does, that tells you that this condition is going to be recessive. Once you know that, you can write your code. And so that means, and I'm going to use F's, lowercase f is going to mean cystic fibrosis, capital F is going to be normal. And we know that because we just said this is a recessive condition. And I'm sorry I write really crappy on my computer screen. My little pointer thing doesn't work very well. Here's the next thing that we can tell. Since this is a recessive condition, anybody who's shaded in, we know exactly what their genotype is. So for every single person that is shaded in, we know their little f. So we can go ahead and write their genotypes in near their boxes and circles, depending on whether they're male or female. From there, we can start to discern who the other, or what the genotype of the other individuals might be. So let's come back to individual 3-1 and her two parents. Neither of her parents are affected. That means we know they have to have a capital F, because otherwise they wouldn't be normal. But these two parents produced a child that is little f. Since she is homozygous recessive, that tells us she must have gotten a lowercase f from each of her parents, and so we know that each of her parents is heterozygous. And so we could go ahead and put that in there. Okay, we know a similar thing up here for this parent. We have a child who has cystic fibrosis. She has a capital F because she's unaffected, but to make that kid her other F must be a lowercase f. Okay, next up, for these two parents, we've got the same story that we did for these guys. Since they are not affected, they must have capital Fs, but they both produced a couple of kids that have cystic fibrosis, so they must be heterozygous. Now we got two people that are left on here. If we look at the parents for those two people, we've got two heterozygotes, these two, that are making this child right here. If we go ahead and do a Punnett square for those parents, so they're both heterozygous, big F, little f, what that tells us is it's possible for them to make a homozygous normal, a carrier, another carrier, and then a kid with cystic fibrosis. Now the reason that we did that is to try to figure out what's going on with this person. We know they have to have a capital F because they're not affected, but here's the thing. We don't know whether or not that person is homozygous normal or heterozygous normal. When you don't know what the second letter is, you indicate that with a little dash. That is the same story for this daughter off over there. We don't know what her second letter is. She might be homozygous or she might be heterozygous. So that's how you use pedigrees. The first thing you always have to do is figure out what the inheritance pattern is. From there, you can start to figure out the genotypes of the individuals that might be involved in that pedigree. Now, to help you get a neater version of what I just scribbled all over on the previous one, this is what we got. So notice all of the people who are shaded in, homozygous recessive, little f, all the way around. Instead of doing the big F dash, they went ahead and indicated that there's two possible genotypes for these two people that we didn't know. The way that I told you is perfectly fine for writing that down now. If you'll notice, we got everybody right up there though. We know who's homozygous recessive, we know who's heterozygous. You can really tell a lot once you figure out how the inheritance pattern is going to work for a pedigree. Now, continuing on with a bunch of definitions, carriers can only exist if you have a recessive condition. If the disease is dominant, there's no such thing as a carrier. You either have it or you don't. There's no other options. So we've talked about Hannah Sames a couple of times in here before. Hannah Sames has a recessive condition. Neither of her parents have it. They are both carriers for her condition. And that means they're both heterozygous. They have a normal version of the gene, and then they have the bad version, and unfortunately Hannah got the bad version from both parents. There was only 25% chance that that happened, and yet their oldest child, that was exactly what happened, was she ended up with both of the bad copies. Now this leads us to inbreeding. Inbreeding is a joke in some cultures. It's taboo in virtually all cultures. And the reason why it's taboo is we know if you do inbreeding, you are more likely to see certain genetic conditions. I don't know how much you guys remember about the Habsburgs in your history classes, but they were the royal family over in Europe. They had people in Spain and England and France and all over the place, and they like to consider their blood pure. And yet they were so inbred that they would end up having to have 20 kids just to have one who actually survived to be like 12 years old. And even then they would be feeble-minded, was the polite way to say. They were super broken, essentially. So if inbreeding continues across multiple generations, disease is more and more likely to increase within that population over time. Next up, we're going to talk about a few different conditions and their inheritance pattern. And actually the first two that I have given you here, they are both dominant diseases. Most people know something about recessive conditions like that cystic fibrosis that we talked about earlier, but there are diseases out there that are dominant diseases. If you just get one bad copy of that gene, you're going to have that disorder. The first of those is achondroplasia. This creates a little person or a dwarf. Now, there's lots of different reasons why a person might be a little person. This is just one reason. I don't know if you guys remember this family. They had a show on like TLC or something a while back. They're the Roll-Offs. Mom has achondroplasia. Now, since achondroplasia is a dominant condition, that means this time the capital letter is the disease. So I'm just going to write dwarf. I'm going to try to write dwarf anyway. The lowercase condition is actually the normal one. And so that's different from the cystic fibrosis that we were talking about earlier. It works kind of different. Even if you are heterozygous, you're still going to be a little person. What that means is, if a little person who has achondroplasia is having kids, instead of there being a 25% chance they have a little person as a child, there's now a 50% chance, because half of, in this case, her eggs are going to have that big A in it and result in dwarfism. Now, this is another good family for us to talk about. The Punnett squares in genetics, it tells you probability. I'm not saying if these guys have kids, they're going to have exactly 4 kids, which they incidentally did, and that the 4 kids are going to work out exactly like the Punnett square shows. Because again, they had a 50% chance of having a kid with a little person. They had 4 kids, him, her, him, him. He is the only one that has achondroplasia like mom. So even though there was a 50% chance, only 25% of her kids have it. For those of you who think I'm being weird right now, he's just a child. Children grow up. He ended up being tall, just like this brother did off over here. Now, to complicate factors, dad is also a little person, but he has a completely different form of dwarfism that is not achondroplasia. That plays with numbers and does other things, but this one we're just talking achondroplasia, so that 50% rule is what applies there. Huntington's disease works the exact same way. It is a dominant condition. Huntington's disease super sucks. It is essentially like Alzheimer's plus Parkinson's all rolled in together. It is fatal, but it doesn't kill you until after you've hit reproductive age. And so very often these people don't know they have it. They have kids, and then it turns out they did have it, and their kids also have it because they didn't know about it until later in life. Now, this is a normal brain. This is a brain of a person who has Huntington's, and you can see there's massive cortical loss. They're missing a lot of brain tissue, and you can see why that would end up killing a person over time. I don't know how much you guys remember. It's been a long time since it's been on the air, but House had Olivia Wilde on it, and she was a character called 13. There was a whole Huntington's story arc for her. Her mother had died young of Huntington's disease, and the question was, did she want to know whether or not she had the condition? Because at present, there's not even a good treatment for this. There's a lot of treatments that are being tested, but there's not really any good ones that slow the disease down, and there's definitely not a cure for Huntington's at this point. So the reason why it was a story arc is, if there's nothing we can do for you, do you want to know whether or not you have it? My 2 cents in here is, it would be beneficial for you to know if you are considering having your own kids. Because if you do have it, again, 50% chance you have a kid who ends up having it, and would you wish that on your child, that they have a horrible genetic condition that's going to kill them fairly young? I totally understand the people who don't want to know, but just kind of consider it from a reproductive standpoint, essentially. All right, next 3 terms, you guys already have all the things defined for you guys, so I'm just going to go through them really, really fast. If you get pregnant, and you know that there's a family risk, that there's certain diseases that you want to find out whether or not your child might have and you're pregnant, the first 2 options that can help you know for sure, amnio is up first. Amniocentesis, they stick a needle through the mom's abdomen. They use an ultrasound to guide it to make sure they don't go in too deep or too shallow. They collect some of the fluid from around the baby. That's called amniotic fluid. There's some baby cells in that fluid. Once we have those cells, we can do tests, like we can do karyotypes to find out whether or not they have Down syndrome. We can do specific biochemical tests to find out whether they have something like Huntington's disease. And so we can tell the mom while she's still pregnant whether or not her child has certain conditions. Now, CVS is very similar. The difference is you run it earlier in the pregnancy. The complication rate is slightly higher than for amniocentesis, but because you're doing it earlier in the pregnancy, if abortion is on the table, it's easier to do that when you do CVS because you know sooner than you would with an amniocentesis. It's essentially similar though. What you do is you stick a glorified pipe cleaner up and you scrape some of the cells from the placenta, and then you run those same tests that you do with an amnio. You just, again, do it earlier in the pregnancy. Ultrasounds can be used to image the baby. You can see like really obvious things like, does the baby have 2 arms and 2 legs? It's very possible for ultrasounds to be read wrong. Probably you all know somebody who was told they were going to have a boy and then it turned out to be a girl. That's because ultrasounds are very grainy and they're kind of difficult to read. They can sometimes be used to diagnose certain conditions, but again, they're not the most reliable thing to find out whether or not your kid has a certain medical condition. All right. What you do with the information once you find out you have a kid that may have some sort of a genetic disease is entirely up to you. What I will mention is this family found out before the birth of their daughter that the daughter was going to have Down syndrome. They used that information to find a daycare that can handle special needs children. They made sure they had speech therapists and occupational therapists on hand that would be able to help their child as she was growing up to make sure she had the best quality of life possible. So that's a completely valid option. The other option is if they have one of the nastier genetic diseases, if you find out early enough in the pregnancy, you can abort the child so you don't have to watch a child suffer through whatever the disease condition happens to be. So you've got options in the United States that are legal. Last few options to complicate things in terms of genetics. Everything that we've been talking about so far, you had a completely dominant allele and a completely recessive allele, and sometimes nature is a little bit messier than that. Using our pea example earlier, if you bred a purple flowered plant together with a white flowered plant, you got purple babies, and the purple babies were the exact same shade of purple as the original parent plant because purple was completely dominant to white. Now, these are carnations down here at the bottom, and instead of purple and white, we have red and white. The complication here is red is not completely dominant to the white allele. So if you breed a red flowered plant to a white flowered plant, well, you get a heterozygote. That is big R, little r, where this is red and this is white. This time, because red is not completely dominant, you can see the white in there too, and what happens is heterozygotes end up looking like something in between. They're pink instead of being red or white. That's what it means by intermediate phenotype. So the problem that I gave you, I wonder if I have something written in there. There we go. So red flowers is incompletely dominant over white. It says to cross 2 pink flowered plants. Well, if they're pink flowered, they must be big R, little r, and they both must be that. If you put those into the Punnett square, the Punnett square is going to look like this right here. So you end up with 1 red plant, 2 pink plants, and then 1 white plant. Again, this is different from Mendel's 3-to-1 phenotypic ratio that he kept seeing when he was breeding his purple and white flowered plants, but that's because he was working with peas, and peas flower color works different than carnations or snapdragons, for example. And so now you get a 1 to 2 to 1 ratio where there's 1 red for every 2 pink for every 1 white. Next complicating factor is sometimes you have alleles that are co-dominant. Codominant is different. They don't blend together to make an intermediate. Instead, you see both examples. So the example that I'm giving you here is, this is a tulip. If you cross a red flowered tulip with a white flowered tulip, you get a flower that is both red and white in different places on that plant. So both red and white are co-dominant to each other. Now, a good example of codominance in people is about blood typing. If you don't know the 4 major blood types, you have type A, type B, type AB, and type O. This one is also complicated by the fact that there's 3 alleles, not just 2. Your 3 allele options are A, B, and O. Here's the issue. Both A and B are codominant to each other. Type O is recessive to both of the other 2 alleles. What that means for you is if a person is type A, that means they might be homozygous A, or they might be heterozygous A, where they're hiding that O in the background and you don't see it. Same thing works for B. You can be BB or BO. Since O is recessive, the only way you can be O is to be homozygous for type O. And then you have that fourth option. It exists because both A and B are codominant. So if you inherit both, it means you're blood type AB. You see both of those things. So here's the problem for codominance in multiple alleles. Husband and wife are having a kid. Husband is blood type O. So we're going to put him up at the top. Okay. And again, sorry I'm horribly messy. I think we've figured that out by now, though. Okay. I'm going to go ahead and fill in his letters with my really messy O's. The baby is blood type O, so what possible blood types might mom have? So here's how we're going to play this. We're going to say that this kid right here is blood type O. Now we can start filling in what mom could be to find out whether or not mom could make that baby. So, mom could be AO, which means she could be blood type A and make that baby. Mom could be blood type B if she's BO and have that baby. Mom could be blood type O and have that baby. The one thing mom cannot be to make that baby is blood type AB. So mom could be any of those three, but she cannot be that in this particular question scenario. Next up, polygenic inheritance I've already defined for you. I've talked about why eye color is a really crappy example to use for genetics. That's because more than one gene tells you what color your eyes are going to be. Anytime you look at a population, a specific trait within that population, if there's a continuum, like think about height. You aren't either tall or short. There's everything in between. That means more than one gene codes for how tall you're going to be. Same for skin color. There's at least 3 different pigments that dictate what color your skin is going to be. So you have a continuum from super pale people to super dark people. That means more than one gene codes for what color you're going to be. The environment can influence some genes, but not all. So for example, her genes did not tell her to be red like that. She went out and she got a sunburn, and so that affected how that was going to be. That said, there are plenty of genetic conditions where the environment plays no role at all. Like, it doesn't matter how much time you spend in the sun or how healthy your diet is or how much you exercise. Your blood type's your blood type. So the environment can affect some genes, but it doesn't have to. Sex chromosomes we talked about in a previous chapter. This person would be male because they're XY. If they were female, both those would be the same length and they would be XX. Now the reason we're doing that is we're about to talk about sex-linked. We talked about them in Pedigrees earlier. Now we're coming in to do an actual genetics problem with them. When you know a condition is sex-linked, it means you have to consider the X chromosome. You can't ignore it. The reason you can't ignore it is guys only get one X. That means they only get one letter. And you can see that here. So let's say this one's talking about fruit flies. Their eye color is carried on their X chromosome. So females can carry 2 copies telling them to make red eyes. The guy, though, only gets one X, which means he only gets one chance to figure out what color his eye is going to be because his other allele is a Y, and the eye color isn't on the Y chromosome. It's only on the X chromosome. The example of a sex-linked problem that we're going to talk about is hemophilia. So this is a very brief pedigree for the Russian royal family. Alexis, P.S. Yes, it looks like a female name, but this is a boy. He was Prince Alexei. Anybody that's half shaded in, it means they're a carrier. This is the first time a pedigree has shown that for you guys. But what I want you to notice is the only person who's actually a hemophiliac is Alexei off down here at the bottom. Everybody else is perfectly healthy. They're just carrying that gene. Once you know that, here's what we can do. Write our code. If a person is X big H, that means they're going to be normal. They make a clotting factor. They can clot their blood. If a person is X with a little h, that means they're going to have hemophilia. And I'm just going to, there we go, hemophilia. Y's do not get the X's and the H's and things. So, if you were going to cross and try to figure out if Alexandra and Tsar Nicholas had another kid, what's the chance that that kid have hemophilia, you would know that, I'm trying to figure out what I can do. I think I need to erase before I can start again. Hold on. Oh, it just doesn't. Oh no, it doesn't like me. Never fails. You try to do technology and the technology really wants to mess with you. Why won't you let me change the color, man? Okay, I'll just try to do my best. So, I'll go straight to a Punnett square. And I'll utilize kind of what I have. So, Y from Dad. Dad was not affected at all, which means he has X with a big H. Now, Mom was a carrier, so she has one X with a big H and one X with a little h. Fill it in the same as always. So side stuff comes over, and then top stuff comes down. Here's what this means. This daughter is perfectly normal. She's not even a carrier for hemophilia. This daughter does not have hemophilia, but she's a carrier. This son, perfectly normal. This son has hemophilia. So once again, 25% chance they have a child who has hemophilia, and that's the one kid they ended up having, which his hemophilia did not help him when he got shot multiple times and assassinated. So hemophilia means you can't clot your blood, so you would bleed to death from just about anything. Alright, so that takes you to the group work. Those can kind of be challenge problems, so you can see whether or not you kind of get the genetic stuff. I'll be posting a quiz in Schoology for you guys to see whether or not you understand the genetics. And of course, when we talk during our next conference, you can let me know if you have any questions about anything. Good luck.